Good morning. This is Daniela Cirillo from the San Raffaele Scientific Institute, WHO Collaborating Center for TB Laboratory Strengthening in Milano, Italy. And it's a real pleasure to be with you this morning and give a talk about the drug susceptibility testing for second-line drugs. This is my disclosure. And those are the topics that I will discuss with you today: the pre-XDR and XDR new definition, the problem of diagnosis MDR and XDR, the drug grouping and the new regimen, and the testing for the new drugs, the quality of the testing, of the current testing, and the challenge. COVID pandemic has highly affected TB case notification around the world. We have observed between 2019 and 2020 almost a 30% decrease in notification in almost all settings. The laboratory area was highly affected, not only because the people had lost the accessibility to the laboratory infrastructure due to the lockdown, but also because the best laboratories were reconverted to COVID work. How do we define the drug-resistant tuberculosis? By definition, strains of MTB complex resistance to any anti-TB drug are defined as drug-resistant TB strain. MDR-TB instead is the definition that defines strains of MTB complex that are resistant to main antitubercular drugs used in the first-line therapy, such as the rifampicin and the isoniazid. If the MDR-TB diagnosis is performed with the gene expert or other molecular test able to test only for rifampicin resistance, those strains are classified as rifampicin resistant. The definition of XDR-TB was changed in 2020. The reason for changing was linked to the fact that the second-line therapy recommended by WHO was also changed. Before 2020, the definition of XDR-TB was including strains that were resistant to rifampicin and isoniazid and resistant to fluoroquinolone and injectable, being these last two categories of drugs the main pillar for the antitubercular drugs for MDR-TB strains at that time. After 2020, we have a new definition for pre-XDR-TB. Those are MDR strain resistant to fluoroquinolones. Fluoroquinolones remains a key drug for the treatment of MDR. And the XDR-TB is defined as the tuberculosis resistant to fluoroquinolones plus one or both the other two antitubercular drugs used for second-line treatment included in Group A, mainly bedaquiline and linezolid. The MDR remains a crisis around the world. While TB is decreasing, MDR number is not decreasing, and we suspect a high level of underreporting linked to the diagnostic capacity. MDR is basically present in every country where there is testing capacity, and we do have three main players in the MDR crisis, and those are the three huge countries that have the largest number of MDR cases, and those are India, China, and Russia. If we want to seriously decrease the number of MDR cases around the world, we need to diagnose the cases in a rapid manner in order to stop transmission, and we need to triage the people for the proper regimen in order to avoid the development of new resistance during treatment. In order to do that, we need to have available fast molecular tests for triaging, such as the rapid molecular test for fluoroquinolones resistance detection. We need to have available confirmatory phenotypic test or appropriate sequencing tests, such as those based on next generation sequencing, targeted sequencing, or WGS. And we need also to have capacity for surveillance for all the new drugs that are used for the regimen. Because although we know that pre-existing resistance to new drug in the population is usually low, we need to remember that resistance to drugs appears in a population of bacteria linked to random mutation that could have hit genes that are target for drug-resistant mechanism independently from the use of the drug. Although this is a rare phenomenon, this happens, and the use of the drug in these cases in the population tends to amplify the drug resistance. And this is what we have been observing with the use of bedaquiline in many countries. WHO has evaluated over time a number of diagnostics that could be used for triaging MDR-TB cases for diagnosis of MDR-TB cases, and some of them for triaging for the proper therapy. In reality, we will see later on that some gaps are still existing, especially when we consider the new regimens that are recommended and the ones that are under trial, that will be able to shorter consistently and make less toxic the treatment. This slide summarizes the current MDR regimen and those that are under trial. So the two main points I would like to make is that we are moving towards a shorter regimen, all oral, and if possible, with a limited toxicity. In order to achieve that, we need to make sure that all the drugs that are included in the regimens have a suitable way to define the sensitivity profile. So, in terms of testing option, what do we have? We have liquid automated culture in MGIT. This is a pretty standard technology, very widespread in the laboratory, is expensive, is quite standardized, at least with the CIRE kit for first-line drug and pyrazinamide, a little bit less standardized for second-line. But when we talk about new drugs, and what do we have? We do have the critical concentration in MGIT for almost all the new drugs. Some of our tentative breakpoint are available only for the liquid culture, not for 7H10 or LJ, but still we are not going so bad. For other second-line agents, such as cycloserine and terizidone, we don't have critical concentration proposed, so we have nothing, as well as for what they were previously called as add-on agent that are now in Group C, for which we really have nothing. In addition to that, we do not have yet critical concentration for Pretomanid. That is a key drug that is included in the BPAL regimen for XDR-TB treatment. Another suitable option to test sensitivity of new drugs is the use of the broth microdilution. There are commercially available plates that they do not contain the new drugs. but the cryptic consortium has used customized plates containing linezolid, bedaquiline, and delamanid to evaluate the distribution of the MIC across a very large number of strains collected from 27 countries, with 19 countries contributing 10 or more samples and 15 countries contributing more than 100 samples. Out of those, more than 12,000 had the whole genome associated information. And that allows us to propose a tentative epidemiological cutoff for those plates. The epidemiological cutoff, or ECOF, is the highest MIC observed when the antibiotics is used on a wild-type population, and is usually defined as the MIC that encompasses 99% of the phenotypically wild-type population. As you can see in the picture, we have used two different methods to evaluate, to estimate the ECOF, both the ECOF finder or the interval regression. In some cases, the ECOF finder and the interval regression were coincident. In some other cases, where the distribution was truncated due to a limited number of dilution on the plates, we have observed some discrepancies. Anyway, the MIC testing is a very good method to evaluate, during the use of the drug in terms of surveillance, how the MIC is shifting on sequential isolate or among the population. The broad microdilution is also the method that the UCAST is proposing as a standard method to develop as a reference method to perform MIC for any new drug. The protocol is published on the website of the UCAST, is not a protocol that is recommended for routine use, but is a protocol that should become a technical reference for MIC determination of antimicrobacterial agents and to be adopted for producing data for evaluation of the drug, to propose the critical concentration for the drug during the process of evaluation of by the regulatory authority of. But when we talk about rapid triaging to start the patients in the proper regimen, we need to talk about rapid tests. We do have some rapid tests that are approved by the WHO. The test that has been used for now many years for triaging MDR cases is the LPA second line. The LPA second line allows a proper triaging of fluoroquinolone resistance, showing mutation and being able to detect mutation to GRA and GRB that are the main determinant. We now have a new test that is the advantage to work on the expert platform, so being more user-friendly, that in addition to the fluoroquinolones determinants, add the isoniazid determinant that for the LPA were included in the first line screening, and targeting the NIH promoter can give us information about ethionamide. In reality, both those assays were designed to perform properly the triage for the old short regimen, when it was key to detect the resistance to fluoroquinolone and injectable. So if we consider those as the only molecular tool, part of the test is already becoming obsolete, showing that there was a disalignment between the development of the test, in particular the expert XDR, that is a new test entering the market right now, with the proposed new regimen for the treatment of MDR. In fact, none of those is able to perform a rapid detection of determinant of resistance to bedaquiline, Pretomanid, linezolid, or delamanid. There are some issues related to the feasibility of that. One thing that needs to be noted is that while we will see that later for bedaquiline, delamanid, and Pretomanid, the situation is pretty complicated, being several genes with several mutations across all genes that are responsible or could be associated to resistance. For linezolid, we only have a couple of genes and a quite well-determined hotspot. So in theory, at least on the expert or on the LPA, if this part is taken apart, this determinant could have. So currently, we are in the situation in which there are no recommended rapid method for the newer and repurposed drug, as the most promising NGS technologies is not yet widely available in resources-limited settings. So we are stuck with the possibility to test by phenotypic method for bedaquiline and linezolid and delamanid. We we don't have tests for Pretomanid, but we will get there later, while we do have good method for fluoroquinolones, both rapid and phenotypic. This slide shows that also in EU country, based on recent surveys that were conducted by two different groups, the situation for testing for the new drugs is not ideal, with few countries performing all 4 phenotypic DST and the majority performing 1, 2, or in some cases 3, in the EU. Few words about the molecular mechanism of resistance to the new drugs to understand what are the challenges to develop new tests. Linezolid resistance, as I've already mentioned, is associated at the mutation in 2 different genes with hotspot. So technically speaking, it is possible to develop a rapid method of resistance detection. Bedaquiline, clofazimine, delamanid, and Pretomanid instead have a much more complex situation with resistance associated to mutation in several genes and with the mutation scattered in the whole gene, and in many cases are not a single nucleotide substitution, but are large piece of deletion or insertion. Is it important to monitor the kind of mutants that develop during treatment? Yes, it is important because allow us to understand the contribution of the single mutation to the treatment failure. This is a case report of a girl that was declared failure in multiple occasions and was treated for a very long time. She developed during the treatment with bedaquiline different mutants, some of them with a very low fitness but a very high MIC that were disappearing naturally, and some others that were instead with a much higher MIC or with a much higher fitness but a lower MIC. The other point is to be kept in mind is that some of the mutations are pre-existent to the introduction of the drug in the population. So when we look back at our strain in the freezer, way before the Pretomanid was introduced for treatment, we saw that we were able to identify strains that had never seen the Pretomanid, but they were already resistant to this drug. Now we have the case of Pretomanid. Pretomanid is a new drug of the Nitroimidazole class, analog of the Pretomanid. Is approved as part of the BPAL regimen and is commercialized with the name of Dovprela. There are no critical concentration officially established and endorsed, although the first paper showing a proposed critical concentration is under publication, has been approved for publication very recently. And there are no molecular screening for testing the mutation in the genes that are associated to the resistance to Pretomanid. There are also information on cross-resistance with the Pretomanid, knowing that there is not a complete overlapping in terms of cross-resistance. The Pretomanid powder is available from TB Alliance and should be made available to the laboratory, at least the reference laboratory or the national laboratory in each country, when the when the regimen will be started. What is really important to be kept in mind is that there is a peculiarity that is coming out with Pretomanid. What you see in the cartoon below is the different lineages of MTB. You can see that MTB is not a clone, although there is not much genetic variation among the strains, but it could be well divided in different lineages that are represented differently in the different geographic area. So looking at the distribution of the MIC with Pretomanid, what was observed is the fact that while the group A and the animal adapted and M. africanum strain have a very low MIC for Pretomanid, the Canetti has a much higher MIC. And among the other lineages, we can see a clearly bimodal distribution, having the strains belonging to the lineage 1 a much higher MIC. So in this case, knowing the lineage will be important to interpret the MIC and the critical concentration to be used for testing. Pretomanid MIC distribution is not the only case that shows a lineage effect. We can see that this phenomenon has been reported for other drugs, including fluoroquinolones. In addition, as you see on the left, the data from Genome Phenotypic Association by the Cryptic Consortium has also shown that the single mutation can contribute differently to drug resistance. This shows the relevance of the identification of the single mutation to understand the capacity to contribute to the process of resistant development. So far I've shown you that we do have phenotypic method to screen for drug resistant to new drugs, not all, because for some we don't have the critical concentration available. We do have the MIC testing, and we do have molecular test. But they all have the limitation because the phenotypic test requires the culture. The molecular tests are not suitable for screening for the new drugs. We can only screen for fluoroquinolones so far. So what is the potential of the next generation sequencing for screening of known genes associated with resistance? We do have two ways to perform that. by target sequencing, and we can do that directly on clinical samples, and we can target up to 14 different genes, 14 different drugs with the current commercially available test. That requires, after the extraction, a step of amplification and then the library preparation. We do have several platforms and kits that will be coming into the market. So far, the one that has been used is the duplex MGIT-TB by GenoScreen. Or we have the whole genome sequencing. The whole genome sequencing has the disadvantage that it requires the culture, but has the advantage that allow to target the whole genomes. So also for future studies allow us to collect precious information. Several works have been focusing on understanding how those tests could be based in a real-life TB workflows. Both tests are not suitable for a complete decentralization. The target sequencing associated with user-friendly platform has the potential to be more decentralizable. The WGS using more sophisticated platform is more for an advanced, intermediate, or central laboratory. The WHO has recently published the first WHO catalog of drug-resistant mutation in M. tuberculosis complex. The catalog is based on the joint effort of the Seek and Treat consortium led by FIND and other stakeholders that have combined high-quality DST data collected around the world with the Cryptic Consortium as a main contributors, high-quality WGS data, a standardized bioinformatic pipelines for the variant detection and annotation, and also a standardized and validated methodological approach for the genotype-phenotype association. The result is the identification of more than 1,200 variants associated with resistance to first and second line drugs with high level of confidence. And in addition, 246 variants that are not associated with resistance. The scoring based on confidence parameters allows to identify and resolve the cases of discrepancies between phenotype and genotype. That has been one of the issues that in the past has been mining for many years the full implementation of the rapid test, with the fact that in many communities and in many settings a rapid test was not acted upon, but the phenotypic test was waited to proceed with the proper therapy. And in the case in which the two were discrepant, either a third of the phenotypic test was used as a standard, making a mistake in many cases. Unfortunately, what is the drawback? The drawback is that this catalog cannot classify mutations to new drugs. And why is that? Because of the few resistant strains available, and because for those strains we are also questioning the quality of the phenotypic data that are available. Does this mean that the genome's data cannot be used to predict resistance to bedaquiline, delamanid, pretomanid, and linezolid? No. We have sufficient research, publication, and data to interpret the presence of deletion, frame shift, insertion, and also SNP in the relevant genes associated to the resistance to the drugs. But now we don't have any mean of associating to them a confidence, a grading on the confidence. The slide summarizes the possible future strategies for triaging based on approaches more rapid than the conventional DST in MGIT. Some of those strategies could be applied on samples, on at least smear-positive samples. Some of them will require a strain. For bedaquiline, that is included almost in all regimens that will be recommended in the future, the best test is to rule out resistance based on lack of mutation in some of the key genes. For linezolid, that is included in the South African short, in the MDR-WHO long, and in the BPAL, there is a hotspot in 2 genes, so the hotspot sequencing could be performed by sequencing, or new tests that could be developed just targeting a small piece of DNA in the two genes. For Pretomanid and the Lamanid, we are more or less in the same situation. So the sequencing of the relevant genes, such as the DDN, FBI, A, C, and B, but we need to understand what would be the cross reaction and how the lineage effect can be influenced. For pyrazinamide, that is becoming a key drug for drug-sensitive TB and drug-resistant TB, then probably the best approach is sequencing of PNCA, or we do have available an LPA based on the LPA genes. Definitely, it is important to establish in any country a program for surveillance of drug resistance, and the WHO has shown in the past that the next generation sequencing, both WGS and targeted NGS, are the cost-effective and comprehensive tool for surveillance of drug resistance associated mutation in a strain population. Going to the conclusion, what is my take-home message? Is that the introduction of new drugs will drive or select resistance. So the new drugs shouldn't be approved without a standard for DST or without a molecular method for triaging the patients. Algorithm for testing should be based on local surveillance. In some places may become soon mandatory to test for bedaquiline. In other settings that could still not be a priority. Testing of samples and isolates should be available and mandatory when the therapy is failing, so at least in one center the test should be available. Triage for fluoroquinolone resistance is feasible with the new WHO recommended diagnostic, but this is the only drug for which we can do an effective and fast triage. The next generation sequencing is a hope, and right now is the only molecular tool to address association to resistance for the new drug. Anyway, the implementation of phenotypic test and sequencing will increase our capacity to classify mutation to new drug in the next catalog. So more data we provide to the community, the better will be our capacity to interpret and to facilitate the development of new tests by the company. And with that, I would like to thank all my collaborators, all of you for your attention, and all the people in the world that suffer because of tuberculosis, their family, the scientists, and the company that are investing in this field. Thank you very much again.